Welcome, ladies and gentlemen, to the second part of my lecture on vaccines. This one is called Vaccine Components and Immunization Technologies, Antigens, Adjuvants, and Excipients. Before, however, dealing with these issues, I think it's a good idea to review what we actually discussed during the previous presentation, and that's summarized on slide number 2. So we were mainly talking about vaccine antigens, the so-called drug substance, and the drug product. And we were also discussing the different types of vaccines, which can be roughly subdivided in several classes like attenuated or inactivated vaccines. Both of these classes can come as whole pathogen vaccines, but also specifically for the inactivated vaccines, specific components from a pathogen, from a vaccine strain, can be extracted and used in the final formulation. So the more modern forms of vaccines are actually split vaccines, subunit vaccines, components, isolated from a pathogen, toxoid-based vaccines, and even more advanced technologies involve recombinant DNA technology as well as live vectored vaccines. And this is briefly summarized in the graph on the right bottom of this slide. Slide number 3, however, tells you something more on vaccine formulations, on all the ingredients, the chemical substances which are needed in order to make vaccines a functional product. And many of these ingredients are called excipients. Excipients are substances which are or need to be added to a vaccine formulation in order to stabilize the vaccine antigen, in order to make the formulation tolerable, to lower reactogenicity, and for many, many more reasons. So the most prominent excipients are, of course, salts, buffer formulations that actually take care that the vaccine formulation maintains a physiological pH, because a non-physiological pH would definitely cause irritability at the injection site and other side effects. And also osmolality, meaning a physiological ion concentration, is a very, very important aspect to consider. So the ion concentration should in principle resemble the ion concentration in the human blood. Anything else would again cause undue side effects. Stabilizers are also important because many vaccine antigens, the protein-based ones, but also the polysaccharide vaccines, are not stable by themselves, at least not for 12, 24 or 36 months, which is the usual shelf life allocated to the different vaccine classes. So typical stabilizers are human serum albumin, recombinant albumin, gelatin derived from pigs or from cattle, bones, sucrose, fructose, sorbitol, as well as urea. These substances can frequently be found in many licensed vaccines. And then finally, water for injection is an important thing to consider. And also we can't go into the very detail. It's very complicated to produce actually water that has the quality and the grade to be used for parenterals. Slide number 4 then deals a bit with adjuvanting systems, which are another group of important ingredients that come together with a vaccine antigen. On the upper left part of this slide, you have a summary of adjuvanting systems which occur in licensed vaccines, like the well-known aluminum hydroxide, aluminum hydroxyphosphate, monophospholipid A, the oil and water emulsions, the saponins, and immune-stimulating nucleic acid motifs. We will come back to that in a minute. But there are many, many more adjuvanting systems which are proposed, which are in research portfolios, and where even non-clinical and clinical studies have been started. So the number of those experimental adjuvanting systems exceeds the number of those already licensed. And that gives you a small hint how difficult it might be to find the right balance between an immune stimulating effect, which is desirable, made by an adjuvanting system, and well, the safety level that needs to be maintained, because even the best adjuvanting system in terms of immunogenicity enhancement cannot be used when it's too reactogenic. This is just unacceptable for vaccines. Slide number 5 actually lists another group of important vaccine ingredients, and these are the preservatives. Actually, they are no longer used nowadays. There are 3 groups of preservatives: thimerosal, phenol, and 2-phenoxyethanol. And they were used actually because both the manufacturing processes as well as the filling of the finished product into vials, syringes, or ampules was not really accepted was not really aseptic many years ago. And that means precautions had to be made in order to ensure that any microbial contamination from the manufacturing process would not increase in the finished product. Such considerations are, however, totally unacceptable nowadays, meaning that the manufacturing processes, the manufacturing premises, in particular the filling lines, ensure that the vaccine which is filled is sterile. So experience shows then that these preservatives are no longer needed. There's one, or maybe there are 2 exceptions. Thimerosal can still be used in multi-dose formulations, and we have 1 or 2 examples from the past pandemic where multi-dose vials were actually formulated with low doses of thimerosal. And phenol is still present in the old smallpox vaccines, which are still stored in some countries in order to have a countermeasure in case variola virus comes back, which is very unlikely. And besides these excipients, there's another group of substances which is found in any vaccine, and these are the residuals from the manufacturing process. And those residuals may be host cell and media proteins from cells that were used to replicate vaccine virus strains. Media proteins may be derived from fetal calf serum or from other proteinaceous additives. There are nucleic acids coming from the same source. There may be traces of inactivating agents like formaldehyde, and also from antibiotics which may be used in the very beginning of the manufacturing process when the cells are grown to large numbers, and also during the incubation phase when actually vaccine viruses meet the cell substrate. But these are normally residuals which can only be measured in very small quantities, and normally or always they do not have any impact on the safety profile of a given vaccine. Slide number 6 now deals with the most complex but also one of the most important groups of excipients or additives added to a vaccine, and these are the adjuvanting systems. A very commonly used definition for an adjuvant is that this is a substance which enhances immunogenicity and overall efficacy of vaccine antigens without causing a physiological effect by itself. And this is why you find adjuvanting systems very often in many vaccine formulations. There's another group of immune enhancing substances, and these are the immune modulators, which are different to adjuvanting systems because such immune modulators like cytokines, for example, they do have an own physiological effect even without the antigen. And this physiological effect very often results in undue side effects. So that's one of the reasons probably why those immune modulators are not present in licensed vaccines, but there are experimental concepts in order to explore what type and to what extent such immune modulators can actually be used. Slide number 7 describes a bit the regulatory landscape. What should an adjuvant do? What is expected? What is should not happen? And the first thing you should consider is there a measurable effect? Is there a medical benefit? And that means that formulations with and without an adjuvanting systems need to be compared side by side in both non-clinical and clinical studies. And the least thing you should observe in the very beginning already of the clinical development program is that the adjuvanted formulation shows a much higher immunogenicity compared to the unadjuvanted formulation, and there should also be a dose-dependent effect for both, for the adjuvant as well as for the antigen. And this is nicely depicted on the graph given in the upper right side of this slide. And that's in relation to an influenza vaccine, which is adjuvanted or not adjuvanted. And the effect is striking and immediately visible. The second thing are the safety aspects. There should be no evidence, or at least only a tolerable evidence that the adjuvanted formulation is more reactogenic than the unadjuvanted formulation. And there's a continuous concern that a substance that enhances immunogenicity towards a vaccine antigen may also enhance immunogenicity against, let's say, cellular tissues, causing probably autoimmunity or autoimmune disease. So this is something that should be carefully considered and should be avoided in order to, let's say, keep the benefit-risk ratio of a vaccine on the positive side. And as I said repeatedly, an adjuvant should not add to the reactogenicity profile of a vaccine, because reactogenicity is something a vaccinee experiences immediately or a bit later following vaccination, and high reactogenicity makes vaccines and vaccination programs unattractive. Slide number 8 explains why adjuvants are needed in modern vaccine formulations. And this has to do with the history of vaccine development, where these whole virion or whole cell-based constructs were over time replaced by subunit or purified antigens, which are much better to characterize and which do actually increase the tolerability profile of a vaccine. And as already discussed previously, an effect that comes in parallel with that benefit is that immunogenicity is actually drastically lowered the purer a vaccine antigen is. These whole-based or whole cell-based constructs actually brought their own adjuvanting systems with them, meaning that the numerous proteins besides the target antigens were actually helpful in enhancing immunogenicity or immune response against this specific target antigen. Slide number 9 describes the expected medical benefits of an adjuvanted vaccine formulation. And as I told you before, the clinical development for adjuvanted vaccines always involves a head-to-head comparison against the non-adjuvanted formulation. And the effect of the adjuvant can actually be seen immediately in that slide. So the immune response against the vaccine antigen is induced much faster and is much higher and persists for a much longer period of time compared to the non-adjuvanted formulation. And this explicit medical benefit or medical effect, together with an acceptable safety profile, has a strong impact on the regulatory decision whether or not an adjuvanted vaccine can be licensed. Slide number 10 describes a scenario where a decision needs to be taken whether or not an adjuvanting system is actually needed in a specific vaccine formulation, in this case for a pandemic influenza vaccine. And these are the lessons learned from the last pandemic, from the years 2009 and 2010, when the so-called H1N1 swine flu virus was suddenly emerging as a pandemic influenza virus. Originally, it was believed that for any pandemic scenario, an adjuvanted vaccine formulation would be needed, but this lesson learned added some additional knowledge to these conclusions. So let's first look at the left part of the slide. This is the surface antigen composition normally found in seasonal strains like H1, H2, H3, N1, and N2. H stands for hemagglutinin and N stands for the neuraminidase, the two predominant surface protections of influenza viruses. And most surprisingly, also this H1N1 pandemic virus had surface compositions that were very similar to the seasonally circulating strains. And that means that in principle an adjuvanting system would not have been needed because most of the individuals globally were already primed through natural contact with these pandemic-like or seasonal H1N1 containing influenza viruses. A bit different is the scenario lined out in the middle of that slide when exposure occurs towards animal influenza viruses like H7N2 and H9N2. In this case, actually, a single dose of an unadjuvanted vaccine would not be sufficient to efficiently prime an immune response efficient against those viruses. So probably 2 or more doses of an adjuvanted formulation would be needed, or an adjuvanted vaccine construct in order to ensure primary immunization against those viruses. And primary immunization is definitely needed, and this is the scenario lined out on the right of that slide when totally novel influenza viruses emerge, like this H16N9 example. Here, an unadjuvanted vaccine formulation doesn't make sense anymore at all. Here, at least 1 or 2 doses of an adjuvanted vaccine construct are needed in order to prime immune systems, since we were previously not exposed to any surface structure comparable to that, and only under those circumstances protective immunity can be achieved against most lethal influenza viruses, like for example the 1918-19 H1N1 or H3N2, which came in 1969, and so on and so on. Slide number 11 gives you an overview on how these novel adjuvanting systems actually work. And very important is to understand the mechanisms of toll-like receptor structures. These are present on the surface of antigen-presenting cells, which can be found anywhere on the surface of our body, under the skin, but also in deeper tissues. And that's collectively what we call the innate immune system. So what you see here is that these so far 9 toll-like receptor structures are able to recognize very simple molecular patterns like double-stranded RNA, like DNA motifs, like lipopolysaccharides, like flagellins, like sugar structures, lipid structures, many more molecular entities as they are present on the surface of possibly invading pathogens. So this is a very complex yet simple mechanism to allow an immediate response against invading pathogens. So that means that the innate immune system is represented by these toll-like receptor structures which recognize simple pathogen-associated molecular patterns. And this is sufficient to fastly trigger an immediate but unspecific immune response against such invading pathogens. And this is mediated by a complex cascade of events occurring on the surface but within immune competent cells, finally resulting in the release of a lot of cytokines which themselves have a strong impact on the quality of the specific immune response, which takes longer to be mounted, but which can then effectively act specifically against a very specific invading pathogen. The next slide, number 12, explains in more detail the relationship between the innate immunity and the adaptive immunity. And as explained in the previous slide, adjuvanting systems, the novel ones, effectively bridge between the innate and the adaptive part of our immune systems. And time is important, and as you see in the upper part of that slide, the innate immune system reacts much faster than the adaptive immune system. So what happens within minutes, hours, or a day as regards the innate immune system takes a couple of days for the adaptive immunity to react specifically against invading pathogens. So it's about these signals that are conveyed by the natural pathogens and which are mimicked by the novel adjuvanting systems that actually drive the adaptive immunity and also have a strong impact on how broad this adaptive immunity is. So to express that in a simple sentence, adjuvanting systems help to multiply the number of signals received by the immune system and the number of signals forwarded to the adaptive immune system. So while an unadjuvanted antigen may provide only a single or few signals, which may result in tolerance or even ignorance or in a low quality immune response, the addition of adjuvanting systems activates the entire immune system in a way that many signals are forwarded which have a strong impact on the quality of the humoral and the T cell in specific immune response. Slide number 13 provides you with a very nice overview on how well structured the so-called novel adjuvanting systems are actually in comparison to the historically used aluminium salts, which, as repeatedly outlined, are in principle a very complex and a very heterogeneous three-dimensional matrix where proteinaceous or carbohydrate-based vaccine antigens can adsorb to. In contrast to that, the novel adjuvanting systems, as exemplified in the middle part of the slide regarding the so-called emulsion-based adjuvanting systems, are very well structured. And both options, the oil in water and the water in oil adjuvanting systems, consist of droplets which can very well mediate between a hydrophilic, a hydrophobic, a lipophilic, and a lipophobic environment, which is obviously the key parameter that allows antigen presenting cells to present vaccine antigens much better than without such emulsion-based adjuvanting systems. And the same well-structured order is found in liposomes, which are also used as adjuvanting systems, where an aqueous core surrounded by a phospholipid bilayer helps again the immune system to mediate between more lipophilic and more hydrophobic environments, again assisting the immune system in finding much better access to vaccine antigens and a much better translation into an effective immune response. Slide number 14 gives you an insight into the molecular structure of novel adjuvanting systems. And as you can easily see from the left to the middle to the right, is that these adjuvanting systems are, just like the vaccine antigens, biological substances because they are isolated from living organisms, from microbial membranes. They are derived from DNA motifs, which are also found into microbial but also viral antigens, or they may be derived from plants. And what is important also here is the well characterized nature of these adjuvanting systems, because the original source, like for example lipopolysaccharide, is quite toxic and innocuous and reactogenic when given in its original status. So the complex work in defining adjuvanting systems actually is to delete during the clinical development or the pharmaceutical development these reactogenic components while the immune enhancing properties are maintained. And that's, for example, the reason why the difference between wild-type lipopolysaccharide, as outlined on the left of the slide, is so different from the monophospholipid, which is actually present in licensed vaccines. And the same is true for the nucleic acid motifs, which are relatively small molecular entities and do not comprise longer sequences of DNA or RNA motifs. And finally, on the right side, the same principle applies for the saponin, the QS21, which is isolated from a plant and which is very well defined in order to keep the immunogenic properties and delete the reactogenic side effects as good as it is possible. Slide number 15 indeed points to the interesting option that the novel adjuvanting systems, alone or in combination, may indeed allow to mount a tailor-made specific immune response against existing and novel pathogens. Slide number 16 points out how GSK, one of the major manufacturers of vaccine antigens as well as adjuvanting systems, actually uses these novel options in order to enhance immune responses against known and new vaccine antigens. So let's start with AS04, which is an MPL-based adjuvanting system, which was the first one ever of these novel adjuvanting systems, which was licensed for a vaccine which is called Fentrix, which is a hepatitis B vaccine, which in combination with this adjuvanting system AS04 gives a much higher and probably a much broader immune response against the recombinant hepatitis antigen used in that vaccine. And this AS04 is actually also the adjuvanting system used in Cervarix, which is GSK's human papillomavirus vaccine, which in comparison to the unadjuvanted antigen gives a tremendously higher immune response. And also another novel adjuvanting system, the AS03, which is an oil-in-water emulsion supplemented with alpha-tocopherol, had a great success story when used in Pandemrix, which is one of the pandemic influenza vaccines used in 2009 and 2010. and which provides a lot of option to protect human beings even from the worst pandemic influenza strains. So there are at least 3 products licensed. There is a fourth one, which is Mosquirix, which also uses QS21 as a further component of a combined adjuvanting systems, and many, many more of these combinations will come up in the future in order to significantly enhance immunogenicity and finally efficacy and effectiveness of existing but also novel vaccine antigens. These trends and tendencies are further summarized in slide number 17. I think I have frequently shown these slides pointing out or pointing to the history of vaccine development. And while the early beginnings of vaccine development started at the beginning of the 20th century, it took about 100 years until the value and the importance of adjuvanting system was finally translated into product development and product licensure. So it's indeed true that these novel adjuvanting systems have been developed during the last 15 years, but the bulk of products has only been licensed during the last 5 to 10 years. But this gives you a very good prospective outlook on how vaccine development will continue. And it's very unlikely that we will ever be able to identify better vaccine antigens, which are more immunogenic. It's more likely that this concept of tailor-made immune responses by developing further novel adjuvanting systems and or by combining them will finally end in a success story that only few have believed that this will ever come through, meaning bridging this so far unsolved gap between the vision of vaccines against many more pathogens and its realization. So we will see how all this continues during the next 5, 10, 15 years. But I'm very confident that many, many more vaccines will come on the market thanks to the development of these adjuvanting systems, which we discussed in this presentation, but also in the previous one. Moving to slide 18 provides opportunity to summarize what we learned during this presentation, but also during the first presentation I gave to you. So we know in the meantime that split and subunit vaccine antigens are highly purified and therefore very well characterized proteins or carbohydrate structures. And those antigens are on the one side very well tolerated, but are very unfortunately no longer as immunogenic as they used to be in their original context. And this disadvantage is efficiently compensated by adding suitable adjuvanting system or adjuvanting systems to a vaccine formulation. And in addition to this enhanced medical effect resulting in a better immunogenicity, efficacy, or effectiveness, there's another advantage which we didn't touch so far, and this is that the use of such adjuvanting systems actually allows a significant reduction of the amount of vaccine antigen needed, which is an important factor because that helps industry to much better exploit their manufacturing facilities without being forced to build new ones because, for example, the demand for these vaccine antigens is constantly rising. So in conclusion, we know in the meantime that a well-balanced antigen and adjuvant ratio results in safe and effective vaccines in principle for any age and risk group, meaning for anybody who might be defined as a target population. So this concludes my second presentation. I hope it has been conclusive and helpful for you. And should there be any remaining questions left, Please contact the organizers of this workshop for my contact address, and I'm more than happy to help you in answering your remaining or further questions. Thank you very much, and bye-bye.